Okay, so this video is going to be a little bit heavy. Just giving you an advance warning about that. Um, I think we're going to start off by just telling you what's going on. Okay, the chapters that you're reading for today, or the parts of the chapters that you're reading for today, talk about electrons falling to lower energy states, and we're going to be harnessing the energy of electrons falling, much in the way that we harness the energy of water falling with a hydroelectric plant, right? And so there's that metaphor of falling water, falling electrons, and that's all in good. Okay. Ultimately, the electrons falling, you might even be able to recognize where we're going with this, is that we're really talking about redox reactions, because electrons are being taken off of something is being oxidized, and the electrons are being put onto something else. Something else is oxidized. And so in this oxidation reduction world, electrons move on to electron acceptors where they have lower energy states. Remember, going from higher energy states to going to lower energy states is the process that releases energy. And that's the energy that we're harvesting. Okay. And so we'll be continuing to unpack that concept. I know that's a heavy bit of verbiage, but it's important for you to get this concept because, I want you to be able to think like a scientist. It's not, science is not about memorizing factoids. It's about understanding processes. And that's what we're doing with our BIO110, right? Along with this, we want to understand, we want to be able to answer, why is oxygen so important in this process? And by oxygen, of course, I mean O2, not the element oxygen. O2 is the thing that we have to breathe in with every breath, because without oxygen, we don't have the opportunity to make nearly as much ATP energy, right? And so going along with this question is a related question, because there's plenty of organisms like yeasts and bacteria that don't use oxygen, and they get their energy from glucose anaerobically, but they don't get nearly as much ATP energy. And so if you understand why oxygen is so important in this oxidation reduction world, you can also understand and explain why the anaerobic processes that use glucose as food are so much less productive. They don't give nearly as much ATP yielding as the aerobic processes do. Okay. And it does kind of come back to a concept that, I mean, this is a little bit, arcane, but it actually is kind of related to something we talked about before. And I want you to remember back to the difference between polar and non-polar bonds. And we said that OH, when you have a covalent bond between O and an H, the O side becomes slightly negative and the H side becomes slightly positive. And that's because oxygen is more electronegative. It pulls on the electrons more, right? And so, yeah, you could sort of say, well, the oxygen nucleus is an electron hog. The hydrogen nucleus is not able to pull as strongly on the electrons, and that's why the electrons are spending more time around the oxygen, right? And so the idea that oxygen is very electronegative is kind of whetting your appetite. I mean, it kind of gives you a little hint as to why oxygen is so important here, right? Because ultimately, we're going to be putting the electrons from our food molecule, from glucose, onto oxygen, right? And so the most stable situation, the final situation with the aerobic metabolism of glucose, cellular respiration, is when the electrons have moved onto an oxygen atom. And when you put electrons on oxygen, when you put O2 and put electrons on, okay, you end up with H2O. H2O is the reduced form of oxygen, but basically this represents the electrons in a very stable state, okay? And so here we're getting to the next phrase that's on my sheet here, which is that you want electrons. When these electrons are on oxygen like this, these electrons represent a very stable state, a very low energy, right? High degree of stability, right? Whereas when you have electrons on glucose, they're not as stable. You'd say electrons on glucose would be a situation of higher energy. Right? And again, our theme here, first law of thermodynamics, is that when you go from a state of higher energy to a state of lower energy, then the energy must be released in the process. And that energy can be used to do things like make ATP energy, or sometimes it'll be used to make heat energy. But we know that the energy has to be accounted for before and after the process. And that's basically what's happening. Now, the electrons, when they're on glucose, are not as stable as they are when those same electrons are around an oxygen nucleus, right? And so having said that, now that we get the idea that, well, electrons on oxygen is kind of like the ultimate state of stability, the lowest point of energy, then it makes sense why O2 is important, right? We have to have O2 in order to put the electrons there. O2 is important because it's the ultimate electron acceptor, right? When the electrons finally make their way onto oxygen to create H2O, we've reached the lowest point of energy. That's the end of the story. We don't go any lower than that, right? So it's kind of like the bottom of the pit. And if we don't have oxygen, we don't have that deep pit to allow the electrons to fall into, right? We have to do something else if we want to get energy, right? And so thinking about the second half of this question, first half of the question, why is O2 important? Well, it represents a very deep pit of energy in which we can throw electrons at the oxygen and have them end up with that very bottom of that very lowest energy point. Right? In the absence of oxygen, now getting at the second question here, why is anaerobic metabolism of glucose so much less productive? In the absence of oxygen, we don't have that strong oxygen acceptor. We don't have that strong oxidizer. There's nowhere for the electrons to fall. That deep pit where the electrons could fall is not there. And so the electrons, if they're going to fall at all, they've got to fall to some lower point that's not terribly that much lower compared to where we're starting off, right? It's like falling off of a cliff all the way down to H2O, or falling off of falling off a very shallow curb, right? You might have some energy released here, but not nearly as much energy as you get when you're falling off of a cliff, right? And so the idea here is that the greater the fall from a less stable state, a higher energy state, to a more stable state, a lower energy state, the greater the amount of energy that's released and the greater amount of energy that could be put into things like ATP production, right? And yes, I will be going to the textbook. Hopefully, having gone through the textbook reading and having had this discussion, maybe things will become clearer for you. That's my hope, at least. Okay. Now, in addition to this general concept, this is kind of like our general understanding. Right. Now we'd add some specifics, and we will be getting more specific in future lectures. But right now, what we're thinking about is, well, at another kind of broad brushstrokes level, we can ask the question, how do we take this huge amount of energy being released, going from the top of the cliff to the bottom of the cliff, going from electrons on glucose to electrons on water, right? How do we harness that energy and do something useful with it, okay? Because it's entirely possible to take the electrons off of glucose and put them onto oxygen. If that happens directly, and I don't know if you're aware of this, but any kind of sugar is completely combustible. Paper is made up of mostly cellulose, which is made up of mostly sugar, glucose, right? And so in the same way that paper, you can light a match to it. If you were to take that same sheet of paper and break it into individual molecules of glucose, light a match to it, it would go up in a very explosive boom, because the for the same reason. The thing is, and you basically end up in the same ending point. If you would take glucose C6H12O6, a whole bunch of them that you have in a sheet of paper, and you add the appropriate amount of oxygen from the air, and it goes boom. Okay, that's the energy that's released, plus CO2 and H2O. The H2O and CO2 are the end products. Okay, we call them waste molecules from the air metabolism of glucose. But what I'm telling you here is that if you were simply to take glucose and light a match to it, you would get a lot of energy in the form of this big blast of sound and heat. Okay, and that's what happens when you combust sugar. The energy is released mostly as heat, okay? And that's because the electrons are being passed directly onto oxygen. And so what cells are doing, okay, the way that we're able to harness this energy and do something useful with it is the energy is not passed directly to oxygen. It's going to go on to intermediate electron carriers. And from there, we're able to do something useful. In particular, we'll be able to capture that energy for ATP production, right? And so another thing that we'll be looking at as we look at the textbook, he talks about NAD+ as an electron carrier. When you put electrons on it, 2 electrons, NAD+ plus 2 electrons will give us NADH, which is the electron carrier, right? And so it's like we could put the electrons and glucose onto O2, and that goes boom, right? Too much energy released all at once, you're not able to harness it, okay? But if you take those electrons and pass them gradually onto some intermediate like NADH, right? And then the NADH can then pass them on to O2 through more intermediates. That gives us a much greater opportunity to harness that energy, right? And so, yeah, it is kind of like the same process. The electrons are falling, but instead of falling in one gigantic boom, we're actually having them fall in smaller steps. And having it fall in smaller steps, we're able to get ATP out of the process, okay? The third thing we'll be looking at as we look through the chapters is kind of getting us back to the cellular biology part of BIO110, where in a cell, in particular a eukaryote cell, where in the cell does this happen? We know mitochondria are involved because we know that mitochondria are associated with ATP production, okay? But the very first step of cellular respiration, glycolysis, is something that's going to occur in the cytosol, which is that part of the cytoplasm outside of the other organelles, right? So outside of the nucleus, you've got the cytoplasm outside of the mitochondria, you've got the cytoplasm, and the part of the cytoplasm that's not inside of one of the membrane-bound organelles is called the cytosol, and that's where glycolysis occurs. And so now you've seen the entire pep talk. Let's go ahead and look in the chapters to see how this appears as you view them in the textbook. Okay, we'll start from the very end, right? Very end, we're looking at the various stages of cellular respiration, right? So, okay, step 1, glycolysis. This is happening. Oh, wait. Let me do this a little bit further down. Okay. This is happening in the cytosol. And so that term hopefully is familiar to us. Basically, this is a mitochondrion here. That's a mitochondrion. And so out here, the cytoplasm outside the mitochondrion, cytosol, right? And that's the location, the cellular compartment where glycolysis, which is step 1 in the process, is going on, right? Right. So the actual metabolism is happening as soon as the glucose enters the cell. We've got step 1 happening in the cytosol. And then after glycolysis, we're going to be seeing this molecule, pyruvate, get drawn into the inner portion of the mitochondrion, right? inside portion of the mitochondrion is called the inner matrix. And that's going to be the place where you have a citric acid cycle, also known as stage 2, right? So mitochondrion has two membranes, the inner membrane and the outer membrane. Inner, guess it's on the inside, outer because it's on the outside. You probably got that, okay? But that means we've got two different compartments. We've got the inner matrix on the inside, and that's where the citric acid cycle is taking place. And then you've got the space in between, the intermembrane space. Which means between the membrane space. And that's going to be the place where we have steps 3 and 4 occurring. Okay? Now, actually, step 3, which would be electron transport. Electron transport is happening on the inner membrane, and oxidative phosphorylation, chemiosmosis, is the part that's going to be taking place as things are moving from the intermembrane space into the inner matrix. And so this is all stuff that we'll be detailing very shortly in like the next lecture. All right, see you then.